welcome to another episode of Nuwairi Darras. This is your host Mahmoud Nuwair. Today we'll be speaking about biochemistry's own RNA and genetic code and what happens with the formation of polypeptide chains and how does RNA help in that, etc., etc. My previous video was in Arabic, but this is a kind of like a continuation to what happens after DNA transcription or synthesis, but this one will be in English though. I may recreate this video in Arabic, so stay tuned. Make sure to like, subscribe, and share, and ask me any questions, leave the comments, and shall we get started? So we're going to speak about RNAs. Alright, so RNAs basically are synthesized, and their main kind of role in the eukaryotic cell is to carry genetic information from DNA to the nucleus and the cytoplasm, and help help with the translation process as well. So there are three types of RNA: mRNA, tRNA, or RNA. So mRNA is called the messenger RNA, and it carries information specifying amino acid of protein to the ribosome. Alright, so the mRNA is a long chain such as we see here, and it has sequences, so kind of every three nucleotides is like an information for an amino acid, and it carries it to the ribosome so it can continue on with the translation process. And then a monocytronic, it kind of means that each mRNA is made for each protein, just like I said. Like each section of the mRNA is for a specific protein, right? And then tRNA is the transfer RNA, and it's responsible for converting the nucleic acid language to amino acid and the peptide language. So it's kind of the main link between the mRNA and the amino acids. It kind of brings in the amino acids and then binds to the mRNA by including an anticodon. So it has an anticodon once it binds, and then helps with the formation of the polypeptide chain. And then the rRNA helps catalyze the formation of the polypeptide chain and splices out introns in the nuclease, and it acts as a ribozymes. So the rRNA is the main structure where all this happens, and it helps catalyze the formation of peptide bonds. During translation, mRNA is recognized by an anticodon. Just like I said, the anticodon comes from the tRNA, and then the anticodon allows the tRNA to pair with the mRNA code. Alright, so we're going to be speaking about four important codes. We got the start codon to start the process. It's AUG. It's found in the mRNA. So this is an essential one. AUG to start the codon, and then you got 3 to stop the codon. You got UAA, UGA, and UAG. Right? I would say the easy way to kind of memorize this or keep in your mind is that AUG, it's 1, starts the codon, and then 3 stop the codon. UUU, and then U stop the codon, and then like a boy or like I will start it. So this is an easy way to remember. It's like, hey, I will start the codon, or like actually A will start the codon. Alright, and then you stop the codon. Next we have two types of mutations. So missense mutation are mutations where one amino acid subs another. So basically when the tRNA brings in the amino acid, it could be a different amino acid, not really related with the anticodon, but it subs for another. And then you got the nonsense mutation. The nonsense mutations relate with the codon encoding for premature stops. So it can be a different codon than these, or a mutated stop codon that stops maybe halfway through or something like that. So missense, it has to do with the amino acids, but the nonsense has to do with like stop codon. It's like, this doesn't make any sense. Why the hell did this stop? Okay, something like that. And then this is the transcription process of an RNA. So the transcription process for the RNA happens this way. So you got the original RNA, and then a helicase kind of opens it up, you know, just like DNA. And then the topoisomerase stops the supercoiling. So helicase opens, topoisomerase stops the supercoiling, and then RNA polymerase locates the DNA region, the promoter region, and then the RNA polymerase 2 is the main factor in the mRNA transcription. Remember that. So the RNA polymerase 2 binds to a region, a promoter region called the Tata box, Tata or Theta box, right? It binds to it, and then once it binds to the Theta box, the RNA polymerase begins to synthesize the 3 to 5. It reads the 3 to 5 strand, or we can consider it a lagging strand, and then synthesizes in the 5 to 3 direction. So it reads the 3 to 5 and synthesizes the 5 to 3 direction, and it continues until the RNA reaches a stop codon, the UAA, UGA, or UAG, and then an hnRNA is formed. So hnRNA is formed. hnRNA is basically pre-mRNA. So before it comes a messenger RNA, it needs to undergo some modifications, you know, before the final product comes out. And then post-transcription, it's turned into mRNA. So post-transcription, there's a couple stuff that needs to happen first. First off is splicing. All right. So you're gonna ask me what is splicing. Splicing is basically a process, a post-transcription process. So after transcription, there are moves non-coding sequences are also called introns. All right, and these introns, they regulate the gene expressions, but they are non-coding regions, and they are removed during the splicing, so the mRNA can mature. And then you got the exons. They ligate the exons together. Exons are the coding region of the gene that they contain the information for the amino acid. Really, introns are not important, so they're like, here, let's just cut them out. We need more space. We need to have more space, so they cut them out, and then the exons are the ones that remain. So this occurs through a spliceosome. So a spliceosome kind of does this part. It cuts out the introns, as we see here, and then you got the exons at the end. And then snRNA or snRNP recognize the 5 to 3, and then the spliceosome kind of cuts the introns and cuts the non-coding sequences. Next, we got the at the end of hnRNA, so at the end of the premature RNA, now it's becoming a mRNA. So what happens? A 7-methylglutamate, a triphosphate cap, is added to the 5' end of the mRNA, or the kind of the N-terminus, to protect the mRNA from degradation, and it's a binding site for ribosome. So ribosome knows, oh yeah, here's the 5 N-terminus, and then we can just say 7 MGT cap, something like that, to like kind of remember it. So the first step after the hnRNA or the premature RNA is that the 7 MGT cap, it's added to the 5 end, and then next we have the 3 end, which we add to it something called a poly A tail at the 3 end of the mRNA to protect it from degradation. And then the longer the 3 poly A tail, the longer the mRNA can survive. So this could be a question on the test. You know, what is an important factor for an mRNA to survive longer? For instance, if we want it to survive longer, you say maybe extend the poly A tail at the end of the 3 end of the mRNA. And next, this is a question that I got, and then I want to just share it with you guys. And then each polymeric molecule is made up of amino acids that has 3 nucleotides, alright? So let's say you have 28 amino acids times 3. So you would calculate that, and then you would recognize, oh, that's kind of like, lower than 87, but no, that's not the case here. Alright, lower or equal 87, but that's not the case here because you forgot the post modification, post transcription modification. These also count as nucleotides. Alright, so you got the stop codon, you got the start codon, you got the poly A tail, you got the cap. These are more than count towards the amount of nucleotides you have. Next we have the translation. So after transcription, now translation. Now it's time to turn the mRNA into a functional protein. Right? So the translation occurs in the cytoplasm. Oh, I forgot. The transcription occurs in the nucleus, and the translation occurs in the cytoplasm. And then there are 3 steps: initiation, elongation, and termination. And then you can just be like, oh yeah, I ET, or I love ET. That could be something. Is that initiation, elongation, and then termination. So initiation is where the small subunits bind to the shine Dalgero sequence in the untranslated region in the mRNA prokaryote. So that happens in a prokaryote. And then in a eukaryote though, the tRNA binds to the AUG start codon. So like we said, there's a start codon and the mRNA, and then you got the tRNA ready with its first amino acid. It's ready to work, it's ready to start the polypeptide. It has the anticodon, it binds to the AUG, so it starts the process. Once it starts, a large subunit binds to a small subunit, forming an initiation complex. These are ribosomal subunits, and kind of related to the rRNA, right, which helps the tRNA and mRNA together. And then this process requires GPT, requires some energy. And then elongation is when the ribosome moves the mRNA 5 to 3 direction and synthesizes a protein in the 3 binding sites, and then it requires GPT. So the mRNA is basically amino acid information to synthesize the protein, and the tRNA is in between the mRNA and the polypeptide chain, so it can keep on increasing the polypeptide chain. So we got 3 sites in the elongation, right? And now we're in the E part. So the first site is the A site. It holds incoming amino acid tRNA complex. So you got incoming tRNA here. It wants to bind to the anticodon. It has an anticodon and it wants to bind to the mRNA. And then the P sites hold tRNA that carry the growing polypeptide chain. You see, this is the growing polypeptide chain, and the peptide bond forms using peptidyl transferase, right? And then the E site is inactivate as tRNA pauses before exiting ribosomes, and tRNA unbinds from the mRNA. So the E site, basically the tRNA pauses, make sure everything is good, and then it unbinds from the mRNA, and then it has released already its amino acid, and it's it's kind of going to get some more, repeating the cycle. So A site, it holds incoming tRNA complex. So oh yeah, now it's this tRNA's turn to come in. All right, and then the P site, it's kind of holds the tRNA that carries the growing polypeptide chain. So there's a growing polypeptide chain. It's your turn in the P site to donate your own amino acid for the polypeptide chain. And then the E, it's like kind of checks in everything, and then it's like, oh, bye bye, you can't unbind now. And then lastly, it's termination. When the 3 codons encountered by the ribosome and the mRNA, the protein releases a factor binds to the termination codon and adds water molecules to the polypeptide chain, allowing the hydrolyzation of the polypeptide chain from the whole structure to form the final chain in the P site. So basically 3 codons, recognized by the ribosome. Protein releases a factor binding to terminate the codon and adds water for hydrolyzation. And then post-translation, we have something called chaperones that assist in the protein folding. You see how there is the primary and secondary and tertiary, and then the 4-step quaternary protein folding. So these chaperones assist in the protein folding. So thank you so much for tuning in. Please let me know if you have any questions, and have a blessed day.